Hello everybody and welcome to this lecture. I'm Cora Thiel and I work at the University of Zurich as a scientific group leader of the cell biology, gravitational biology and biomechanics group. Additionally, I am the coordinator of space life sciences at the UZH Space Hub. In this lecture today, I will talk about how microgravity can be used for biotechnology production and applications. This slide gives an overview of the different microgravity platforms. The drop tower allows experiment times of up to 9 seconds. On parabolic flights, 22 seconds of microgravity time can be achieved, while on suborbital flights and sounding rockets, the microgravity time is in the range of several minutes. If longer exposure times, like days or weeks to microgravity, are needed, the International Space Station or even CubeSats are the platforms of choice. What are the environmental conditions in low Earth orbit? The space environment is a very special and hostile environment where you find radiation, extreme temperatures, vacuum, and microgravity. For this talk, I will only focus on the aspect of microgravity. In microgravity, various phenomena known on Earth are significantly altered, like density gradients, gravity-driven convection, reduced surface and interfacial tension, buoyancy, hydrostatic pressure, and sedimentation. These characteristics are also the advantage of microgravity. We can perform investigations of features that cannot be manipulated on Earth. Microgravity alters many observable phenomena in physical and as well in life sciences. On the right side, you see a cell that is attached to a surface, sensing and reacting to different stimuli, like for example shear stress or gravity. The cell and its organelles are highly structured. For example, the chromosomes in the nucleus are not randomly distributed, but each chromosome has its very specific position. The nucleus is connected via the cytoskeleton to the cell membrane, and environmental stimuli, like changes in gravity level, can be directly transmitted to the inside of the cell. This leads then to a rearrangement of the chromosomal positions and to global alterations in gene expression. A big advantage of cells growing in microgravity is that they tend to aggregate and grow three-dimensional and form tissue-like structures that resemble much more the physiological conditions inside the body compared to a 2D cell culture. Therefore, microgravity is very well suited for biotechnology production and applications. In this lecture, I want to focus on 3 topics: biomanufacturing with pharmacology, protein crystallization and antibody production, disease models and aging with tissues and organs on a chip, and third, tissue engineering and regenerative medicine, including stem cells, 3D bioprinting, and organoid production. What are the challenges on Earth? The pharmaceutical drug discovery process is long, complex, and very expensive, and there is a high failure rate. A study usually starts with several thousand compounds, and then, when entering the clinical trials, only a few compounds are left. And only 10% of the compounds that enter the clinical trial stage advance to FDA approval. In the picture shown on the left, the study starts with 5,000 to 10,000 compounds, and after 5 years, only 1 compound reaches FDA approval. Also, disease and aging research using cell culture and animal models is very cumbersome. Research requires long experiment times, and the knowledge transfer from models to human is often very difficult. Also, in the area of tissue engineering and regenerative medicine, there are big challenges. An organ shortage exists, and patients often have to wait for many years for an organ donation and transplantation. However, our population is getting older, and there is an increased need for organ donations as well as for cell therapy, and together with this for improved disease models. Organoid production is a rapidly growing field and could be the solution for these challenges. However, there are limitations of organoid productions on Earth. Due to the gravity environment, three-dimensional growth of organoids is impaired and scaffolds are required as supportive structures. Production in space allows to circumvent those problems. With respect to pharmacology and drug discovery, protein crystallization is an important step to identify protein functions as drug or drug target. The environmental conditions in microgravity allow substantially improved crystal growth, and compounds could already be identified with an increased drug efficiency, with an increased serum half-life and increased concentrations. Also for disease models and aging research, investigations in space are beneficial. Tissue and organ-on-a-chip platforms represent new opportunities to grow tissue-like structures in 3D and to expose these structures to different substances and conditions. These platforms have been already used for studies of cardiac dysfunction, kidney and bone function. There is currently a big effort to address the wide field of tissue transplantation and personalized medicine with organoids. I would like to give you here a short overview about what is an organoid and what are the possible application fields for organoids. Organoids are multicellular 3D structures that derive from stem cells. They develop into complex structures made out of multiple cell types and show self-organization as well as specialized organ function. This means they are organ-like. Organoids provide an enormous application potential and can be applied for drug discovery, toxicity testing of substances, substitution of animal testing, regenerative medicine, and many more. By using microgravity as a tool for tissue production from stem cells, it is possible to produce efficiently and robustly organoids without additives or matrix structures normally required on Earth, enable early testing of potential drugs on human organoids, reduce costs of preclinical tests by a factor of 5 to 10, and we can start research on personalized drugs as well as research to repair damaged organs generated from personal stem cells. During our pilot study in 2020, we developed the necessary protocols and executed successfully our space experiment where we sent 253 samples to the International Space Station. Human stem cells derived from bone marrow were cultivated with specifically defined cell culture media that drive the stem cell development into tissue-specific organoids. In our first attempt, we wanted to generate 5 different human tissues. The analysis of the samples showed that in each of the 253 cell containers, we obtained complex organoids. This means our project was 100% successful. And what is also very important, the organoid structures produced in microgravity are 100% natural. That means no additives or matrix structures are required for the formation in space compared to Earth. We therefore developed a highly efficient and robust organoid production system. As you can see in the picture on the left side, the organoids reached diameters of up to 1.5 millimeter, which is an ideal size for transplantation with respect to vascularization. On Earth, organoids produced with artificial scaffolds are much smaller and usually have sizes of below 0.5 millimeters. This slide gives an overview of our results. For all tissues, you see a clear difference between the 2D ground control with no or only minor differentiation and the ISS samples that show exactly the features of the textbook references which are displayed in the last row. You might ask yourself if research or production in space is expensive. This picture shows that the launch costs are constantly decreasing with the long-term goal that by 2040, the launch of 1 kilogram will only cost tens of dollars. This slide gives you some examples of research and production applications of pharma companies in space. It includes drug testing, crystallization of therapeutic antibodies, disease models, and lab-on-a-chip technology. What are the future perspectives for biotechnology production and applications in space? Recently, the ISS National Lab made a research announcement for in-space production applications like tissue engineering and biomanufacturing. Additionally, NASA is taking more steps to support new commercial space stations in low Earth orbit. The company Axiom Space plans to build a commercial space station between 2024 to 2028 for research and manufacturing. With this slide, I want to summarize and conclude my lecture. I showed that the new space environment will enable easy and affordable access to space, that microgravity is a unique environment with almost endless possibilities, and that space economy is of high scientific and economic value with respect to biomanufacturing, disease models and aging, as well as tissue engineering and regenerative medicine. With this, I would like to end my talk, and I thank you for your attention.